Thank you very much, and good afternoon to everybody. In these days, I am talking to people who are not biologists or molecular biologists, but now is laser experts. And I have to say to you that Friday, I gave a little talk because I get a literature award, and this. So now I get a Libri Award I received because my book was sold the highest number last year for the non-fiction category. So I also tried to hear, Ferenc showed all of these fancy pictures, which was real pictures about laser and other things. And this is the fanciest picture I could find, because otherwise this is imaginary picture. I have to say that you can make RNA research in your kitchen table, and the budget of $5,000 or $1,000, you don't need any fancy things, no vacuum. And the closest I get to laser research is that at the Biological Research Center, when I worked there in 1985 to 78 to 85, Joschka CG, he was doing laser research, and he was next door to me. And sometimes, it was big sign that not to disturb, but when it was open, the door, I just peeked in, and I could see dark, and there I could see some beams, and he was sitting by himself very quietly, and I could see some kind of magnifier things was put there. I didn't know what was, and I imagined that I would never want to be a laser scientist. So I stay in my room and did mRNA research. So the messenger RNA was discovered in 1961, and it took like 60 years when eventually, as a form of vaccine, of COVID-19 vaccine became, approved. And if you follow the literature or news last week, and other vaccine, the respiratory syncytial virus, just those who are above 50, there is a lot of young ones, so not for you. Who is older than 50 is already approved by FDA. So a lot of things happen, and I will talk about what. But actually, well before 61 discovering mRNA, people already realized that the RNA can carry information, because what they did with the tobacco leaf they took, and they brushed in phenol-extracted RNA to the leaf, and it was infected. So they realized that the nucleic acid, which is RNA, carries all of the information, and it can be a virus. So people said, what the hell is all of this protein is doing? But we know that they are protecting and helping to spread. But it was in the 50s. And they repeated similar experiment in animals. They injected, and realizing that the pure RNA can carry all of the information and create a virus. So, but how this RNA is it present in our body? One question scientists ask, that here we are, all of the chromosome is in the nucleus, and all of the protein synthesis is going on in the ribosome, and they could not find the DNA in the cytoplasm. So it took long years. They tried to identify that who carries this information there. And I just mentioned one Russian scientist, when they did Spirin and colleagues, they come up with a great idea. They selected 20 or 30 different bacteria, which the GC content was that, I don't know, GC means anything for you? GC content was very high, so that, and then they thought that if they isolate the RNA, they can prove that the RNA was made from the DNA. And that was a great idea. And so they did, and you can see that the GC content was very high in some of the bacteria, but when they isolated the RNA, voila, there were no difference. So I had once in the wall in my laboratory that it was a quote from Leonardo da Vinci. Experiments never fail, only your expectation. Because they didn't know that they isolated total RNA, and it was mostly ribosomal RNA, which is identical in the bacteria. We know today, and we laugh at them, but you know, this is how science, we always try to figure out something, do an experiment, and we get a result which we don't expect. So anyway, I told you that RNA, messenger RNA, was discovered in 1961. These two papers, back to back, about this discovery, and both mentioning that it is a very labile molecule. And now finally we understood that the central dogma of life is that there are DNA, and RNA is made and goes to this ribosome, makes protein, and we are made from protein, or the protein is enzyme, makes lipid, other things, and that's who we are. And so it was very exciting. I have to say that this paper was published in 1961, and several of the authors are still alive. So meaning, if you work on the field, you can expect long life. And scientists couldn't make mRNA. What they did, you know, they discovered quickly that there is some kind of long poly-A tail is there, and other scientists get the idea that we can use it for purification. So sometimes in 1972, they invented this oligo-DT, purified the mRNA, and this helped other scientists, mostly chemists, look at the other end of the RNA where they found this cap structure, and Furuichi was actually in Japan who first discovered that there is a cap on this RNA, and if you get the mRNA vaccine, yes, it was a cap on it, and because they also showed here, without cap, there is no translation. So you need that cap there. And I might mention that in 1975, many papers came out in Nature. So if you worked on this field, you get a good publication, and grant is funded then. But by the end of year, Bernard Moss demonstrated there are enzymes which can do this capping mechanism, and it was from a viral enzyme, and if you get the Moderna vaccine, they used also the same vaccinia capping enzyme. The BioNTech incorporated co-transcriptionally a different manner. So these were mostly chemists looking at the RNA. They cannot synthesize, just isolate, and others try to look at more function. I just described here one. You know, the precursor of red blood cell is reticulocyte, and it is full of beta globin because, you know, the red blood cell is full of hemoglobin. And so what they did, they isolated and put in a lipid, and they delivered to a primary cell in culture. And then this cell, this spleen cells wouldn't make globin protein, but now it did. So it was 1978. Yeah? So it was those who think that vaccine research started just in 2020 is mistaken. A lot of people worked on this field. I was not talking about what I was doing, but in 1973 I came here, Szeged, and the reason is because I want to go to the University of Szeged, because in the same year, the Biological Research Center, 1973, was opened. And that was my dream, that I will work there and I will discover something. Yeah? I didn't know what the scientist is doing. My father was butcher, and I did not know any scientist up until, you know, I came here. So I tried to I loved plants and genetics. The only opening was in the lipid lab. So the grease stuff is not very exciting, but I was looking forward to spend the summer at the bench. Professor Tibor Farkas told me that, Kati, rest of your life you can spend at the bench. I will send you to a field trip and go to Szarvas. There is this Fischer Institute, and please collect some water fleas. So, you know, important things, you have a dream, you think you are at the bench discovering big things, and then you are in the water and collecting something like water fleas and make a little experiment. And that was actually basic of my first publication, came out 81. And of course, here I collected fat from fish. And if nobody's from the industry, I can say that, if you feed the fish with corn, it's not really the healthy fat there. So try to get fish from the Northern Sea captured. Anyway, so I hardly could wait to go back and finally, and the bench research, and Ernő Duda and Eva Kondorosi work in the same biological research center, biochemistry, and they came with the idea that they want to have liposome, and they approached the lipid team, because this is from lipid, and they wanted to deliver DNA to the cells. And, I was like 22 something. I just thought, great idea, and the problem was that we were behind the Iron Curtain. And that's why every time I hear about sanction, I always think about fellow scientists behind. So that's what happened to us. We couldn't buy, with the 2 Deutsch mark, was this forced reaction that Judith Forsch described in 42. How to have this reaction of phosphatidylserine ethanolamine. Anyway, Ernő found this paper and jumped on the bike, and he came back with a bucket of co-brain. And so it was a good thing that my father was butcher, so I was ready to act. And then, purify out with acetone the protein, then the ether and all of these things. And this was the time, that we had no fume hood, just open window. So I inhaled so many chloroform, ether, other things that sometimes when I don't remember something, I blame on that it's solved. Anyway, at the end, we could deliver the DNA to the cell, and this we published. And so what is important for the student, you do an experiment and you never forget, and then it was always in your mind. Because one day when Jenő Thomas walked in the laboratory, and Jenő is an organic chemist, and he said that he has an opening for a position for doing a PhD. This is how it was at that time. I don't know whether it is different. And I said, oh yeah, so RNA, I will go there. So from this, you can obviously see that I am not a visionary. I just follow the flow. Anyway, and Jenő told me about the cap. He worked actually with Furuichi. They get a publication. And he sent out, different cap analogs to Aaron Shotkin to New Jersey. And so I already learned from mRNA has a cap. And then he told me that I have to synthesize this molecule, which is a short RNA molecule. And I worked together with János Ludwig. That I met him. He's a chemist student, and when Hétvezért, we collected grapes in the spring or the fall, and then that's what I met him. And now we are working together again, and we are working even today. We get patent together, many publications together. So I am saying here, be nice to the people sitting next to you or working with you, because you need their advice. So this small molecule was thought to be responsible for the interferon effect, that naturally we are making this molecule, oligoadenylate synthase, when we have double-stranded RNA infecting us some virus, and then we produce this molecule, and this will bind to other enzyme, and the idea was that it chops up the viral RNA, and this is how it exerts antiviral effect. And if you follow the literature on COVID and during the pandemic, they identified these are the two enzymes, oligoadenylate synthase and RNase L, that those people who had very low level, they get seriously ill. And those children who get ICU, they also had a polymorphism, which resulted that level was very low. So anyway, we get support from the industry. I don't know, I was népköztársasági ösztöndíj I got, and I was, you know, kind of valedictorian and so on, and I get Academia Award, and I get a stable position, and somehow I had a child, and somehow I was a temporary position, and one day it was ended. So I don't even know how it happened, so I don't blame anybody, but I had to leave the country, and I had to go to America. And if you read my book, this is the laboratory, had no windows and low budget, you know, no lab coat, nothing, and a lot of cockroaches. But anyway, we worked on this modified nucleosides, and Professor Suhadolnyik actually wrote the basic book on modified nucleosides. And we started to develop also 2' 5' link molecule. This is a cordycepin is an analog, easy to make 2' 5' because there is just 2' hydroxyl there. And actually that was part of my PhD thesis here at in the center. Anyway, we were working and we couldn't make this small molecule good antiviral. So what happened is we turned to double-stranded RNA, which was known to induce high level of interferon. Interferon means interfering with viral infection, but it is very toxic. So what we did, we introducing some mismatches. And this was called Ampligen. Other superiors made a company, and we started to treat HIV patient. We are in 1986, and we treated 10 patients. Unfortunately, their interferon system is compromised, and so we couldn't help them very well. But I might mention the major advantage of being America at that time was that, you know, we could go to lectures. So, you know, there were no telephone, no iPhone, no internet, nothing. If you want to know what's going on, you might have a Nature or Science, and every week you learn what happened 2 weeks before, or you go to the lecture, and that's what we have done. So different lectures. Anyway, you heard that I was so small RNA, double-stranded long RNA, I did, but I did not talk about mRNA yet. Because the most important thing happened in 1984. By that time, you know, I was just about going to America when they figured out how we can make mRNA in a test tube. The test tube is really a tiny tube, I have to say, like a couple of microliter reaction I use. I was first time shocked when I was at CureVac, and I've seen the 50 milliliter reaction, and I have to say that Pfizer is doing 60-liter plastic bags. So that things increased there, but anyway, now we could make synthesized in test tube, and those were scientists from Harvard University. Douglas Melton, Paul Krieg were first described in 1984 how to make mRNA in a tube. Because now that if you have the genes, you just can copy it and you can have an RNA. And I sent a letter when, you know, with stamp on it, kind of. So because there was no email at that time, and asked Paul Krieg that whether he could send me this construct because I want to make mRNA, and kindly he said that we have even a better one, which T7 promoter, and he included that one. This is how friendly the colleagues were to each other when it was in the late 80s. I have to say, you know, that at that time was revolutionary things. I mean, Tech polymerase was 1989 was the molecule of the year. So the PCR is just coming in and isolating RNA. I mean, Chomchinsky introduced the cow-tropic agent. Up until that, to make RNA was really a pain. So I would hug him one day, you know, if I meet him, because it was made my life so much easier. Anyway, we made, you know, they made this human beta globin interferon encoding RNA, And they had a capping enzyme from Bernard Moss, I just mentioned. He figured out how to isolate from virus, and then they injected to frog oocyte. And Zsolt and others who are at my age, you know, that if you walk in a biology lab, they have a terrarium with a frog. And this was not for decoration. They had big eggs, and then it was easy to inject to them. And that's what these scientists did, injected this mRNA which they produced, and when they cultured it, that supernatant contained functional human protein. So this was 1984. So I was also excited. I already get the package from Paul Krieg, and I was yes, I lost my position actually at Temple University, as 4 times it happened in my life. No guarantee to get a Nobel Prize if you 4 times you were terminated. But without all of these things, I wouldn't be here today. So Elliot was interested. He's a cardiologist and a week younger than me. So I immediately, when he hired me for a faculty position, expecting that, you know, soon I will get my own funding. He waited for 7 years and never get. Anyway, he provided, because he was interested in this urokinase receptor, and this receptor is functioning if all of this glycosylation, I don't know whether you can see from there, different glycosylation in there, it is GPI linked, and so many decoration is needed. And what was interesting is I cloned this urokinase receptor, I made the mRNA, and then it was like, it was functional. So the cell knew all of this what to do. And then with Elliot, we decided that it would be good for something. Maybe for older people can give bone marrow, because we can extend the tip of the chromosome and with the telomerase and edit the template, and then it will be useful something. I also did a lot of play. Elliot did, you know, they did bypass surgeries, catheterization, other things in clinical department, you know, molecular biologists can do anything. And then I was having different blood vessels, mostly not human, but animals, and I put RNA like dripping through, and 15 minutes later, protein I could measure. So it was like, it is feasible because they take out usually the blood vessel from the legs, and it is in their hands, and you know, in a couple of minutes, they can put an RNA there, which has beneficial effect. So we thought that it will be useful. We were not the only one. A lot of people were thinking about that mRNA will be good. And so the 90s, you can see, they already using mRNA for therapy. They made vaccines, influenza vaccines, cancer vaccine, and other things. I usually mention at that point, you know, that to follow the literature is kind of easy, once a year on mRNA. And in these days, you know, like every hour you have more than that. But anyway, the interesting part was that these scientists hardly published anything anymore. So I was very excited. Floyd Bloom was the editor of Nature. He's a script. They did injected vasopressin to the brain, and they could treat the animal like 2 weeks, had no disease. And I decided that I will do mRNA coding for therapeutic protein. But somehow all of these scientists did not publish, except Eli Gilboa, who also made a company, went all the way to a phase 3 trial, but due to futility, they had to stop. So anyway, later life, you know, I meet people and they respond when I email now. And then, you know, ask why you didn't do more experiment, and turn out that they couldn't get funding. But because Elliot paid my salary, so I was not worried that much. But most of them, you know, problem was that this RNA was unstable, very short period of time, just very small amount of protein was made, and it was not enough for therapeutic. You had to figure out what kind of disease it would be sufficient. So what you could see for years is that constant improvement of the mRNA and the formulation, because you want to formulate, you have to protect this RNA, and then you want the formulation to stick there and have to enter to immune cells, and then when it is an endosome, you want this that capping wrapping material release the RNA a little bit, but then let it go to the cytoplasm. And there are many different formulations. There were like protamine, which is in the sperm cell. It sticks so well and never released RNA, so that lot of experiment had to be done to find the right wrapping material. So this was the story there, but now that Elliot left cardiology, I was saved by David Langer, who was a medical student in Elliot's lab. And if you have medical students in your lab, you know that they know everything. But I still suggest to you to be very kind and nice to them, because David convinced the chairman, he was already a resident, and convinced the chairman that neurosurgery needs a molecular biologist. That was me. And then I get this corner. This was my laboratory for 17 years. This was, this one-person laboratory, because I couldn't get grant. You can see low budget again, no lab coat. Only recently, when I became famous and the university wants me to show up with the logo. But anyway, we did all of this research and try to figure out what kind of mRNA product we can make. And so that's what I am telling you. You are on one field, and if you are another person, another field, and you respect and you educate each other, even two persons can come up with some novel ideas, like David said that, I was talking to a person who has subarachnoid hemorrhage. I'm talking to that person, blood is in the brain, and he said 2 days later he will be dead, because all of these major blood vessels will constrict and he will die. And then he said that with some hocus pocus, I don't know, physician know that he said that I can reach this area of the brain, and what we should do, I said I can make a nitric oxide synthase mRNA, so in that case not the protein, but the protein is an enzyme which makes nitric oxide, which is a couple of millisecond half-life, but locally, if it releases there, maybe we can help this patient. So that we started to, I cloned and we started to test out. We were in Chicago, different places where model animals were there. So with the ice bucket and with the RNA, I was traveling here and there. We tried to find, to show in an animal model. And this was, David left. He got an offer at Lenox Hill, and if you watch Netflix, there is a series, David Langer is playing there in the Lenox Hill or New York Emergency. It was last year was the series. So anyway, and he established the largest neurosurgery department in the United States from scratch. Yeah? So he's very talented guy, and he helped me. Although he left, he came back every year when, I was ready to kicked out from neurosurgery, and he made sure that I stayed there. But my salary was $50,000, and neurosurgery department is always the richest. You remember Christmas party, catering, infectious disease department, they have to bring the food, so there were different. Anyway, but so they still paid my salary. It wasn't, penny for them. But one day I really ran into Drew Weissman and at the Xerox machine. And for the young one, I can say this is a machine, it's a big one. And then we copied from journals the article. Then we put to the file cabinet. We put numbers and reference. So it was quite complicated that time. Anyway. From 2002, everything is, I save digitally, so if the progress would be better, I never see through Weissman because I wouldn't go to the machine. But anyway, he was there copying, and he worked in the infectious disease department, and the neurosurgery was a different department, but because in cardiology, I remember the code for the Xerox machine. I used that, and then so that's how it happened. We start to chat, and I like to brag. I told, I am an RNA scientist, and he wanted to make a vaccine. Prophylactic for HIV patient or prophylactic or therapeutic. So he said he already tried DNA, peptide, other things, but not RNA. And I was, brag about that I can make RNA. And he gave me the template. And then I made GAG mRNA for HIV. And what was important that he came to Fauci's lab. At that time, I didn't know who he was. And then he cultured this very specific human dendritic cells. This is a professional antigen presenter cells in the immune system. Anyway, so as it was critical, and both of us needed for this study, and I have to say we really were very low on budget, and we did all of this experiment, shoulder to shoulder, really, because no technician. And so anyway, we did the first experiment, and he was very happy because all of this important marker was high up in these human cells, but we also measured high TNF-alpha. And TNF-alpha is an inflammatory molecule, and I was there to save the lives of the stroke patient, and had last thing you want to inflammation. And that was the question we asked them, why the RNA is inflammatory? Where is this TNF-alpha is coming? I thought that I make the RNA which is inside the cell, and then came to the thinking, because everything is curiosity-driven. We didn't set up, we will do non-immunogenic RNA. We just thought that why it is. And maybe all of the RNA is inflammatory. And so by that time, I was working on RNA 10 years, so I have a repertoire of, in the freezer, things, different RNA isolated here and there, because I always needed control. And for this experiment, tested out. The purple shows the RNA, which I made, which I thought that the best. And then turned out that all of the RNA I isolated were less immunogenic, less in TNF-alpha was induced. And when we looked at closer, we found that this tRNA, this is transfer RNA, was not immunogenic at all. And we already knew, especially, Suhadolnyik lab, we just did everything, modified nucleotides, that 25% of the nucleotides in the tRNA is modified. And that's got the idea that maybe modification is the reason why it is not inflammatory. And they are, okay, now just we have to prove it. But how? I'll just show that because you are not chemists, just to scare you a little bit, yes? There are more than 100 different modifications in the RNA, and most of them is in our body. We always made RNA from the four basic nucleotides, and we have enzymes, and we change it. And these changes is introduced. And the importance is not fully understand what enzyme is doing, really not known. And so you just couldn't order from Promega to send an enzyme, and I can introduce this. But I knew 2 modification was introduced by an enzyme, and this was Tamás Kis. And he is from University of Szeged. We lived in the same dormitory, and I am telling you, it's not enough to remember student that somebody was handsome or good soccer player. You have to follow what they are doing when they leave the school. So Tamás discovered that how 2' methylation and pseudouridylation occurring in the ribosomal RNA. And because I was nice, so I called him up and asked him to send me these kind of components so I can modify the messenger RNA. But of course, it was not that easy at that point because it wouldn't work in vitro. So I had to call up János Ludwig. We collected the grapes once upon a time, and I told him that I have to, instead of doing how nature is doing, with enzyme changing, I have to buy nucleoside triphosphate, which is already modified, and, you know, crossing my finger that maybe the enzyme will incorporate. And so I asked him where the hell I can buy nucleotide triphosphate, which is already modified. I insisted only that those which is present in the human body. I don't want any foreign things there. And he suggested to me the company, which later became billionaire, the company, not we, but, you know, because everybody was purchasing the modified nucleoside triphosphate from them. And 5 of them incorporated. So we have mRNA, which now contain modified nucleotides. And the big question was, of course, that whether it changed anything. And when we put the RNA into isolated dendritic cells, which actually drew isolated from the human blood, what we found is that some of them were still inducing inflammatory molecule, but some of them was not inflammatory. And when we look at closer, we could see that which was not inflammatory, then the uridine was modified. So it's a big discovery. Exactly 2 science paper came out that U is activating Toll7/8 and Toll-like receptor 7 and Toll-like receptor 8. That's what you happen when you make major discoveries. But we at least knew that how to change and not to activate. So we have these modified things, and I have to say this we also produce that Toll-like receptor 8 by itself. Just the uridine, the nucleoside, can dimerize and can activate. And it was published later, as well as, you know, mRNA. Naturally, your mRNA in your body also has pseudouridine, so it is not a foreign. Even prior to that, it was known that tRNA and short RNA has, but it also has the mRNA. Again, it was 10 years later was known. We didn't know at that point. Anyway, because Drew wanted to make vaccine, I want to make therapeutic protein-coding RNA, and so it is important that it will produce some kind of protein. And the surprise was that pseudouridine-containing RNA, when we deliver to different type of cells, even primary cells, we get 10 times more protein. And we didn't understand that instead of the uridine, the conventional RNA, it is so much better. Why nature didn't introduce that? And I don't want to bore you with those 2 years which I tried to understand all of these things. But I can mention that whatever cells I tried, and of course, working neurosurgery, sometimes I have to touch neurons. So I showed that, you know, neurons, RNA can be delivered to the neurons, and they express, this is GFP encoding mRNA was delivered. And when we deliver to any kind of cells, mRNA in a culture, like 99% of the cell picked up and translated. And so it was very powerful. And what we have done usually in the United States, you file a patent and establish a company that you have never heard of, but because we couldn't get the patent for our own company. That's happened in other countries, hopefully not here. I don't know whose, you know, universities here, so that it would be the best to give to those who made the discoveries. Anyway, I was already in between all of this termination. I was also demoted from my faculty position. I didn't even mention that. And wrote a small business grant that how this mRNA, if it goes for erythropoietin, how it could be used to treat anemia. And so did the experiment, and we injected this nucleoside modified RNA with a commercially available complexing agent, and we could see that even 4 days later, we still could measure the protein. I have to say that we did experiment, when we injected, 30 minutes later, we already could measure the protein. So when you inject an mRNA, instantly translates, and if it is contains pseudouridine modification, for example, it will be like 4 days translation ongoing, and it is functional. The protein made from this RNA functional, increase the hematocrit value, because the erythropoietin is a doping drug. And then when weekly injected, we could maintain high level of hematocrit. And the animals were very happy, jumping up and down. And if we had a bike race, probably they would win. Anyway, and then it was not inflammatory. So this was 2012 when my last grant was rejected, and chairman said that if it is not, then I have to go. So I ended up in Germany. I have to say that at the age of 58, my biggest accomplishment I felt was that I got 2 job offers, one from Moderna and one from BioNTech. Now though you are smiling, but BioNTech and Moderna was not known. BioNTech didn't even have a website. So anyway, I said, Germany, I said yes, and then there were no fancy building like that, just a campus, and then I joined forces, and we started to further improve the RNA, optimize the sequences, purified many, many different things. One important thing I did there for 9 years working in BioNTech is that everything what we have done at the bench at University of Pennsylvania, I have to repeat on a way which is scalable for industrial manner. And when realizing that the RNA is not pure, it has double-stranded RNA contaminant, had to work out. And again, this was a lot of work when finally we figured out an HPLC purification, which is not usable for industry, so we had to come up with a different purification procedure. But it, in the light blue, shows that how well it increased the translational, and this is log scale, improved the translation, so purification. Anyway, all of this hocus pocus, finally, the little protein, short period of time, now that by 2013 we had a lot of protein, long time, and the RNA was ready for prime time. And you already could see clinical trials started. I mean, CureVac in Tübingen was established in 2000, and later 2009, it was for 8, BioNTech was established. 2010, it was Moderna, and everybody was gearing up for run a clinical trial. And so I didn't talk about why is this mRNA so important? I mean, you have to know that more than 100 different product is FDA approved, and if you are a physician, any of you know that how expensive the protein base, and you have to choose which patient to give, because antibodies and other things are really very expensive. But when you use the mRNA, you can inject to the patient will make the medicine. And then you can make a polyvalent injection, because you can mix RNA coding for many, many different proteins, because they are always from the 4 nucleotides, and they will not stick to each other. If you would try to mix the protein, one is hydrophobic, you would get an aggregate, and it would not happen. Then much cheaper even if the final product is protein, the development is much cheaper, because you can make RNA very quickly. I just say one thing here, what I did in BioNTech, for example, because I was in charge of the protein replacement program. And we did a bispecific antibody. This antibody can see the immune cells, can see something on the surface of the tumor cells, bring close proximity, and then half-life is very short, like 2 hours. The patient who gets a bispecific antibody get a pump in their arm, and then every two days they had to fill up the pump with the material. But here we injected the mice, in a humanized mice here, and then it was eliminated, even established large tumor. This is already very successfully in clinical trial, human trial. While I was working, of course, in Germany, I keep a good relation with Drew Weissman and Norbert Pardy, who worked in lab at the beginning. He worked with me. It's, Norbert, grandfather, and my father work in the same butcher shop in Fischl-Szalás. So that was, we established good trust there, and we put in charge that he will make these vaccines. And what was interesting is that this vaccine was very similar. It was against Zika. We are in 2017, not HIV is the problem, not corona yet, Zika. And right before the Olympics, you remember that. And then he made RNA, which coded for this envelope protein, and it was 1-methyl pseudouridine containing, and it was formulated with aqueous lipid nanoparticle. What was interesting is that when he injected to the mice, like 30 microgram, or injected 50, 30 microgram to the monkeys, who not working with monkeys 4.5 kilo around, so much larger. And it was the same amount of RNA was sufficient. And it was a surprise because when DNA was used as a vaccine, unproportionally, so larger the animal was, unproportionally more had to be injected. So finally, in a human trial, when DNA was used in Africa, milligrams of DNA was injected. But here it seems not. It is the same is enough, and you are already aware that for corona vaccine also, mice 30 microgram, monkey 30, human 30 microgram was sufficient, and generated protective immunity. And from this one, you know better than maybe me. What was the timeline when Yongtsen put out the sequence information as a Shanghai virologist, and he put it there in January 5. I think he's told me. I got an award with him, and that time I talked to him, and he said that I put to the gene bank, but nobody paid attention. And at Holmes, his friend from Australia, he asked him to put the Twitter to tell the people that the information is there. So you see that you need every part of the technology. Even you need Twitter to look at what is in the gene bank. And so when the people learn what is the sequence information for this new coronavirus, 150 location people started to make vaccine. Information. If it would happen 20 years before that, everybody would expect, not everybody, but at least who want to make vaccine, a little package from Wuhan with the little virus in it to start the vaccine development. But by that time, you just can order genes. Tomorrow the genes arrive. You can make an RNA in 1 hour. You can inject. And that's how it is. Of course, scaling up is a challenge. So you can see that Moderna already injected the first volunteers in March. And the trial ended before, end of that year. Of course, skeptics said, oh, this is the first time messenger RNA vaccine was used for infectious disease, but those people did not attend the mRNA yearly meeting because they would know that actually CureVac much earlier already tried to develop vaccine to protect against rabies. Moderna ran flu, avian flu, 2 Zika viral vaccine trial and CMV trial already, and those were lipid nanoparticle formulated. It is just, 100, 200 people, not 200 million. And so beside that, when we had first time, 2013, we had the mRNA conference, and there were, the panel of experts, I was not even part of it, but, the industry and everybody was there. And, nobody mentioned that infectious disease will be the first which will be approved. Everybody thought that VEGF-A mRNA, which was used by Moderna already in phase 2 trial, where they injected naked mRNA into the heart, and they could see that those patients who underwent elective bypass surgery, the heart performed much better. And so that's everybody thought that will be the treatment. That others, like Intellia, a company, started to use Cas9, which is a genome editing enzyme, and then they deliver the mRNA to interrupt a very toxic protein, which, these people who get this transthyretin amyloidosis, and they are dying after the diagnosis like 5 years, and they could interrupt it in their liver, this toxic protein production. And this is already close to approval. So there, it seems that even the dream of gene therapy may be solved with mRNA. I still have a little time to tell you about that what's going on today, because more than 250 messenger RNA is in human clinical trial. And most of them against viruses. I listed some of these, the influenza, the respiratory syncytial virus, now it's going on for the younger one, and HIV, the HIV virus, the Zika virus, and herpes simplex virus, and that was initiated from University of Pennsylvania. EBV, CMV, Nipah, which is in Asia, is a major problem, or monkeypox, recently you heard about. And then there are bacterial infection. There is tuberculosis and the Borrelia, which cause Lyme disease, and like malaria, which also ongoing and sponsored by Gates Foundation. And of course, in the news more you can hear about cancer vaccines, and those are individualized or share tumor antigen, and not only vaccines, but antibodies, which I mentioned, and also there are T cells, which again generated with mRNA. The T cell therapy is very expensive because you have to generate viruses to transfect the T cells, but now there are also possibility to use transposons, and that can deliver the gene. And I mentioned acute diseases. There are genetic diseases now that Moderna announced they are a rare disease company, and they have several trials which is already past phase 2. So they might be approved, and many allergies like peanut and dust mite allergies, they also use the mRNA, and so it is a lot of things is coming on its way, and I just can quickly show you that all of these years, because, okay, 1961, I did not read those Nature papers. I couldn't speak English, read English, and I was 6 years old. So, but later on, I was part of this process. And I remember at the beginning how we begged people to send gene clones, for example, because the clone was not available. Later, companies formed, and we could purchase clone. And then came the RT-PCR cloning. So when we had a cell line which contains that particular protein, we know that RNA is there, we isolated, and RT-PCR TA cloning, we could make it. Later, we could order the clone, and now that if we needed some modification, we just can make custom synthesis. The same way improved how we will make RNA, that the cap, for example, first we just incorporated the simple cap 0, later enzymes became available, we could make poly-A tail, we could make cap 1, and so on. And of course, if you don't care about cloning and RNA, you just order it, if you have money. And finally, the delivery, which I mentioned. liposome already in the 60s, they have, but not using for RNA, but then, different commercial product, transit lipopectin was available, and protamine, which I mentioned, it sticks very well to the RNA, and it was not very useful, but now that there are lipid nanoparticles. So there are progress constantly ongoing, and companies are formed. These are just the beginning companies, but now all of the large company has RNA unit. And now what is left for me is to inspire the next generation. Some people doing, not me, other people are, this is Weissman and that's me. And we are, inspiring others. Other people wrote that. I wrote this paper, this book about my life. It will be published in 13 different languages, 9 already out. So if you want to know how to survive the harsh environment in the laboratory, just read it. I have give you a lot of advice. And finally, I mentioned that I like to brag, and if you wouldn't know, I am the mother of a 2-time Olympic champion, and I used to go to her races and award ceremonies, usually introduced as Susan Mom. And when she is coming now to the ceremonies, she is Kati's daughter. So it is, and thank you very much.